name's Norelle. I'm from the Microbiological Diagnostic Unit Public Health Laboratory, which is based at the Doherty Institute in Melbourne, Australia. So my poster is called Game of Clones, and it's about the sequential emergence and expansion of multi-drug resistant and extensively drug-resistant strains of Shigella sonnei in Victoria. So as a background, Shigella is an enteric pathogen, but it also wears a few other hats. It can also be a sexually transmitted infection, which is what we've seen a bit more recently in high-income countries. And so we undertake routine genomic surveillance for Shigella in Victoria, so that all isolates that are found in the diagnostic labs are sent through to our laboratory for surveillance, and we undertake culture and whole genome sequencing. And what we've seen over several years is that we have lots of different clones that have come and gone over time, and the clones differ by the risk factor type. So here in this large phylogenetic tree, we can see a few different clones. So we've got in blue the ones associated with MSM, so men who have sex with men. And we've seen those with varying degrees of drug resistance, from not very resistant through to multi-drug resistant, and then extensively drug resistant, where we have resistance to all of the most commonly used treatments, including ceftriaxone, that's resistance mediated through extended spectrum beta-lactamases or ESBLs, and azithromycin and fluoroquinolone. So these are ones that we really worry about. So we've got 3 different MSM clones that have come over different time points. And in red, we have the more diverse clones that are associated with travellers who are returning from endemic countries where we know Shigella causes a lot of diarrheal disease. And then in here, we have a recent foodborne outbreak that was at a music festival. And that was unfortunately an extensively drug-resistant Shigella sonnei clone. Again, with resistance to all the first-line antimicrobials. So that actually required us to speak with hospitals and clinicians to make sure they were aware of that. And then over here we can see a pattern of clonal replacement. So although the clones are a bit different for the different risk factor groups, particularly in the MSM group, we've seen clones come and go over time. Part of that is likely driven by resistance, but it's not always entirely clear what that is from. And then lastly down here, we can see that our travel strains are very diverse. A lot of them come from Southeast Asia due to travel patterns in Australia, but the others can come from all around the world. So that's my poster. Thank you very much. So I'd like to thank my co-authors. This was largely work from Jake Lacey and Sally Dougal, who are a bioinformatician and epidemiologists in our laboratory, as well as the rest of our laboratory crew and the diagnostic labs who send us the isolates, and the Department of Health Victoria, who work with us on this and provided data on risk factors. Thanks.